Well, hello once again. This will be our last lecture in this series that we've been doing on the cells of the human body. And relatively short one. We're going to cover the organelles. As you recall, we had covered cell structure, membranes. We had looked at various transport mechanisms through which things cross back and forth in and out of the cell, across that membrane, and many of the processes and laws that govern the movement of these materials. So today we're going to finish up that chapter, and we're going to do that by looking at the organelles that make up the cell. We're not going to spend a lot of time here because frankly they don't have a lot of relevance to what we're doing. It's not that they're unimportant, of course they are, but in terms of what we're going to study, we're going to be operating at a slightly higher level of organization. But some of these, as we'll go through, I'll point out, we will pay attention to, and they will come up from time to time. So let's begin. So this is going to be the organelles. The organelles that we find inside the cell. And let's make sure we understand what the definition of this term is. So these are specialized structures within cells that have a characteristic shape or that have characteristic shapes and that perform specific functions. Okay. So if I drop that and add the S there, it makes a lot more sense. So specialized structures within cells that have characteristic shapes and that perform specific functions. So let's begin with the largest and most prominent of all, and that would be the nucleus. And hopefully by now you do understand what the nucleus is. So the nucleus, of course, is the structure that when we look through the microscope at a typical cell, it's something that we usually, if not always, see within that cell. And at this point in the class, if this were a face-to-face class, you would have now had that experience, and I could draw upon that experience to talk about that structure. So for the time being, we're going to just pretend like that has happened. So let's give this a little bit of a description. So these are typically oval shaped. So typical oval shape. So sometimes they are not, of course, and particularly when you start looking at white blood cells, they have very unusual nuclei. And in fact, such unusual nuclei that can be used to identify specific types of white blood cells. They are very prominent, easy to see. Okay. And there is usually only one. Now, there are exceptions to this, exceptions within the human body. So, for example, skeletal muscle have many. They're multinucleated. Red blood cells, on the other hand, have none. So there is some range, but typically we're going to find one nucleus per cell. Okay. They are bounded by the nuclear envelope. So the nucleus is defined, the borders of the nucleus are defined by a structure we call the nuclear envelope. And this is a double membrane. And here we need to really make sure we understand what we're talking about here. A double membrane. And here I think I will go ahead and bring in a nice little picture for us to look at to emphasize exactly what it is that we mean when we say there's a double membrane. And so here's a nice image that we can use. And if you look, and I should do that in English, there we go. And if you look carefully at the side picture here, so they're taking a section of this. This is the entire nucleus, obviously. And they're going to take a section of that and blow that up into detail. And you have one lipid bilayer right here, and then you have a second lipid bilayer. So I have two lipid bilayers here, two separate and distinct lipid bilayers. So that's what we mean by a double envelope. And since we're looking at the image now, we can also see that there's these structures called a nuclear pore. These are protein-bound openings through which things pass in and out of the nucleus. But again, I'm emphasizing the fact that we have two membranes here. And so let's quickly draw a little detail in our notes to get that down, so we have a sense of that. And I'm going to go ahead and I'm going to put that here is one nuclear envelope. There is the second nuclear envelope, or I should say second membrane. So I have one membrane here, and I have one membrane here. Okay, so double membrane. And here is my first one. One lipid bilayer. And then two lipid bilayer. And as we saw, there are structures that span both membranes, creating an opening through both of them. And these things are the nuclear envelope. Oh, excuse me, the nuclear pore. So I have nuclear pore. Okay. So that's what we are looking at when we look at the nucleus. Okay. So we'll go ahead and nuclear pore is a protein bound opening into the nucleus, and it allows the passage of small molecules and let's pull that out so you can see that. There you go. That was the passage of small molecules, such as, for example, ions, as an example. Okay. There is another structure that we often see, though it's not as clear sometimes as I would like, as I was teaching this class, and this is called the nucleoli. And if we come back to that image we were just looking at, we can identify that. So here's the nucleolus. So that's the singular form, the nucleolus. This is a dark region that we see inside the cell, inside the nucleus, I should say. Here is a actual photomicrograph, a transmission electron microscope image with that prominent structure in place there. Now, once again, I will mention that had we been in a face-to-face situation, and we would have looked at, by now anyway, frog skin to look at simple squamous epithelium as an example of simple squamous epithelium. And typically, this structure shows up for some reason in those cells, and I would have pointed out to the class, and and had you look at that, then when we had this lecture, you would have had something to think back on and to relate to. So this structure within the nucleus is the site of ribosome construction. And we're going to talk about ribosomes. Hopefully you have some idea of what they do. They are involved in the synthesis of proteins in the cell. So ribosomes are a combination of proteins, RNA, right, together. Okay. As you know, the nucleus is this place where we store the genetic material. So the genetic material is stored, and what we're talking about, of course, is DNA. And DNA comes in two forms. So two forms. And the first form is what you typically are going to see if you're looking at a cell, and this is called chromatin. And chromatin is what we call diffuse DNA. It's all spread out. It's not coalesced and packaged together. When we coalesce and package the DNA together to create this second form, which is identified as chromosomes. So you often have heard that term, I know. When you're talking about chromosomes, of course, you're talking about the genetic material, the DNA, but it's in the condensed form. And that only occurs when the cell is going through some cellular division, such as mitosis or meiosis. So it's not commonly seen very much, anyway, in your typical examples of cells. All right, so that's the nucleus. Let's move on. And since we already talked about it, let's talk about next the ribosome. So ribosomes, as I said, these are for protein synthesis. Without these, there is no protein synthesis. You cannot build proteins without ribosomes. So they allow you to create the proteins that you're composed of. And they are composed of two subunits. And believe it or not, these subunits are referred to as the large subunit. I don't know, somebody was asleep at the wheel. This seemed like a perfect opportunity to pull in some Latin or Greek, but they didn't. So large subunit and small subunit. And when you put these together, you have a complete functional ribosome. So some of these ribosomes, some ribosomes, are free and unattached to other organelles. And others are attached to a type of organelle that we're going to talk about very shortly here called rough endoplasmic reticulum. So let's take a look at a little picture here just to give us a visual of what we're talking about. So two units, a large subunit and a small subunit, come together to make a functional ribosome. Some we find free, some we find attached to structures such as the rough endoplasmic reticulum. All right, so here we are. Here's the large subunit, small subunit. We hook them together and we have a complete functional ribosome. And you can see here the example that they're giving is ribosomes that are attached to the rough endoplasmic reticulum. Now, they are also found free within the cytoplasm, but many of them are attached to the rough endoplasmic reticulum. So there is your ribosome. And that's really all we need to be concerned about in terms of that structure. And since we have been talking about this endoplasmic reticulum, let's go ahead and complete that discussion. So the next thing on our agenda will be endoplasmic reticulum. And let me just take this. So the next structure we want to talk about is endoplasmic reticulum. Endoplasmic reticulum. All right, so a general description of what this is an extensive network of membranes that form flattened sacs or tubules within the cell. Okay. These structures have a name. The flattened sacs and the tubules are referred to as cisterns. The endoplasmic reticulum is an extension or extends off of the outer layer of the nuclear envelope. So it extends from the outer layer of the nuclear envelope. And remember, there are two membranes that make up the nuclear envelope, and the outer one forms these large extensions that we are referring to as endoplasmic reticulum. Okay. In a typical cell, more than 1/2 of the membrane surface in the cytoplasm is composed of rough endoplasmic reticulum, or excuse me, endoplasmic reticulum of both types. Okay. We get two forms. The first form is called rough endoplasmic reticulum. Rough endoplasmic reticulum, or sometimes shortened to be rough ER. So that is a recognized abbreviation of that longer term, rough endoplasmic reticulum. So, rough endoplasmic reticulum is characterized by flattened sacs upon which ribosomes are attached. And this is all about protein synthesis, obviously, since we have the ribosomes present. So if a cell has a lot of rough ER, it's involved in the manufacture of a lot of proteins one way or another. So this is typically where I like to drift back into my childhood, because when I was growing up in Oakland, this is in the 50s. Yes, I am that old. Um, I would walk to school, and Emerson was my elementary school. And on my way to school, on the corner, was a candy store. And this was an old-time candy store, kind that you don't no longer see. So at that time, in most cities, in a lot of cities, I should say, the groceries and all of these kind of things that you would normally buy at a Safeway or Lucky's were being sold by small mom and pop grocery stores. In this case, it was a candy store owned by a sweet little old lady. And just to walk into that place and smell the candy was amazing. But one of the types of candy she sold were these strips of papers, and upon those strips of papers were little gumdrops, were little sugar drops, I should say. And you would buy a roll of paper, and then you would just attach one little sugar drop at a time and eat it. I have seen those in other stores, but they are not the same, believe me. So something has been lost there. So that's rough ER. And the second form is the smooth endoplasmic reticulum. And again, this can be abbreviated as smooth ER. Okay. And here we're talking about tubules. Okay. It often extends off of rough ER, so it's an extension of rough ER, and is composed of tubules. And tubules is just like it sounds. It's small tubes. Tubules, okay? There are no ribosomes in this case. And of course, this is all about the synthesis of lipids, be they phospholipids, steroids, fats, what have you. Smooth ER is all about the synthesis of lipids. So if there is a cell, for example, if there's a cell that's producing a steroid hormone, you'll have a very extensive smooth endoplasmic reticulum. And the rough endoplasmic reticulum will be there, but very much reduced, as opposed to a cell whose primary job is to produce a protein-based hormone, for example. You would see extensive rough ER in order to build the protein. So let's take a look at that. So here we are. And here's the nucleus. There's my double membrane. And you can see there's an extension. There's an extension out of the membrane. It starts forming these flattened sacs. And as those little purple dots supposed to represent ribosomes. And of course, what's happening is the manufacture of proteins. The proteins end up being inside the cisterns, inside the sacs. And in that way, you're controlling the process rather than just building something out here in the cytoplasm and letting it drift off to wherever it wants. You're containing it within the sac. And as we'll see later, we can then ship it to another structure in the cell. And so we can start to add things. We start to modify that protein in ways that we might need. And then, as you can see, coming off of the rough endoplasmic reticulum is this whole structure that looks like a bunch of tubes, and that's the smooth ER, and that would be involved in the synthesis of lipids. So the processes that happen in the rough ER and the processes that happen in smooth ER can be related to each other. In other words, the proteins can be being modified with lipids, but not necessarily. So this process could be entirely different than the process that's taking place here in the rough ER, even though there is that obvious connection between them. And then further on down in the page, you can see This is again a transmission electron microscope, and here you can see the rough ER, the flattened sacs, and these little dots that you see there, those are the ribosomes that are attached. And then you get into the smooth, which has this more tubular appearance to it. So that's the endoplasmic reticulum. Interestingly, you're going to run into that. We're going to run into that structure, the endoplasmic reticulum, in our study of skeletal muscle cells. Now, it's not going to be called endoplasmic reticulum, but that is, in fact, what it is. It's a modified form of the endoplasmic reticulum, and I will just leave it at that so that you're all excited when we get to that and wondering what I'm talking about. So, a little nugget out there to keep your interest. Okay, the next thing that we're going to talk about is the Golgi Complex. And this consists of about 3 to 20, again, flattened membrane sacs. And your typical shape, and I'm going to go ahead and draw a nice little picture for us so we can picture that. Your typical shape, I'm trying to figure out what color pen I want to use. I'm going to go with red here. Hopefully that can show up quite well. So, oh, there we go. So your typical shape is going to be something like this. So here's 1, 2, 3. You get this kind of, I've only shown 3. There could be up to 20. Okay. Material. There is a orientation to this structure. So there's the so-called cis face, which is on this side. And then this is my trans face here. Okay. And what is going to happen is that you're going to bring in materials from here. And as we modify and change those materials, they're going to move from one to the other and then out in this direction. So the movement of materials through this structure is from the cis face to the trans face. And it just depends. And typically then you'll end up at maybe, say, the plasma membrane. Next stop, plasma membrane, as you leave the trans face of this structure. And again, it just depends on what's happening there, what's happening in the cell, as to how many sacs and what the processes are that needs to happen there. So it's a processing and packaging organelle. So modification of the original material is happening inside the Golgi complex, and it's packaged and sent out someplace. Let's take a look at an example of what we're talking about here. Okay. So let me get rid of this. All right, here is my rough ER. I've manufactured some protein. So now I'm going to squeeze off a transport vesicle. I've captured that protein in that vesicle. It's going to enter on the cis face, and things will happen to this. We'll have modification, changes, clipping this, adding that, whatever needs to be done. And then it's going to transfer through a vesicle to the next structure, and eventually it's going to be placed either in a secretory vesicle such as this, and then exported out of the cell. That would be exocytosis, or it might be actually be part of the membrane. It might be one of the proteins that we talked about that were part of the membrane, or it could be something that needs to be digested, as we see here. So some cellular material debris that needs to be digested and we'll talk about the lysosomes, which is what their job is for digestion. So basically, we're receiving materials at one end. As we move through these sacs, we're modifying them in some way, and then we're transporting them out to their final destination, whatever it might be. All right, so we talked about this, the lysosome, so we might as well go ahead and bring that up into our notes. So let's talk about that. This is our lysosome. And these are membrane bound vesicles produced from the Golgi Complex, and they contain powerful enzymes. And this is basically the digestive system of the cell. Whatever is going to get dumped into these vesicles is going to be digested. So whether it's worn out cellular material, or even if in the case of phagocytosis we've captured some bacteria, we'll merge that vesicle that has or is containing the bacteria with a lysosome, and that will be digested. So it's a way to digest and sort of process material inside the cell that has lost its usefulness. So digestion of extracellular material as well as intracellular material. And we contain those enzymes inside a vesicle, because if that vesicle ever ruptured and the contents were let loose into the cell, it would basically digest the entire cell. So this is something that cells are going to do, obviously, is to protect themselves from these very powerful chemicals that they sometimes manufacture. We will also see this, or I should say you will see this in 40C when you're looking at the digestive tract. So many of the proteolytic enzymes, these are enzymes that digest proteins, are packaged up inside cells in such a way as that they are basically non-functional. And then they're released outside the cell where some process makes them functional, and then they can start digesting your meal. So this is another example of how your body deals with dangerous chemicals inside the cells. So these protein enzymes are very dangerous because they're going to digest proteins, and they don't care where those proteins are, whether it's your meal or something inside the cell. And so the cells that are responsible for producing these very destructive enzymes package them or produce them in a non-functional form, and then they become functionalized once they're dumped into the digestive tract. All right, well, let's move on to one organelle that you will be talking about quite a bit. So this is actually a very important one that you will be running into, and this is the mitochondria. So the mitochondria are, a nickname for this is the powerhouse of the cell. Okay? And it produces ATP from the oxidation of glucose. So all those little pinging sounds, if you're hearing them, are you students sending me emails. So that's you trying to get a hold of my attention. All right, so powerhouse of the cell produces ATP from the oxidation of glucose. And this is why we have lungs, to bring in the oxygen to burn the glucose. So oxygen is a very reactive gas, and if we didn't need it, we would want to keep it out of our body. But we do need it in order to produce ATP efficiently. So that's what the mitochondria are all about. And it's a very efficient process if we have oxygen available to when we burn glucose to get molecules of ATP out of it. Let's talk a little bit about how they're constructed. So mitochondria are a double membrane, two membranes, just like what we saw in with the nucleus. There's two distinct membranes. So let's go ahead and let's get a quick picture of that and label some of the parts. So here, blue will be our outer membrane. So this is our outer membrane. That one membrane there. Okay. And then we have an inner membrane. And this one is folded quite a bit. We have this folding. And I might not have wanted to do all this, but I've started, so let's continue. It's actually kind of fun. There we go. So we have this double membrane. And so obviously this is called the outer membrane. And this one is called the inner membrane. And this center part is referred to as the matrix. And what we get is cellular respiration is happening on this surface right here. So the processes that produce ATP are happening on the inner membrane. So why is it folded? Why is it folded? Well, it's folded because by folding that membrane, we get a larger surface area, which is something that would be very valuable if you're making a lot of ATP. So we get this folding, and that folding increases surface area, and the structure becomes more efficient in the manufacture of ATP. Okay. So this is fascinating by itself, but there's another fascinating part of this story, which is that when we looked at this, let me back up. So there was a biologist, a female biologist. Her name was Lynn Margulis, I believe that's how she pronounced her last name. And she wrote a book. In which she hypothesized that many of the cellular structures that we are seeing in eukaryotic cells were actually prokaryotic cells that were engulfed by another cell and then took up permanent residence within that cell. So this was a very controversial idea. And she took as her sort of case in point the mitochondria. So she was asking herself, why is there two membranes? What would produce a two membrane bound structure? And she hypothesized that, well, if this were some sort of bacteria and it got engulfed by another cell, that cell would form a vesicle around that structure. And so you would produce this double membrane. Now, typically when that happens, this would be digested or destroyed. But her idea was that at some point in the distant past, the advantage of this staying alive within the cell, well, there was either a mistake in that it wasn't destroyed, and then it provided some advantage to the cell. And then over time, it simply became totally integrated into that cell and became part of that cell. And she really took a lot of flack for this idea. Until people started to investigating that theory and looking for evidence to support her. She had her own evidence, of course. She just wouldn't come up with an idea like this without any kind of evidence. And it turns out there's quite a bit of evidence, particularly with the mitochondria. So let me just outline a couple of things that make that hypothesis supports that hypothesis. First of all, the mitochondria contains its own DNA. So we find DNA just as we would expect to find this if this had been a living organism at one time. We find DNA in the mitochondria. Now, all of the genes that are necessary, I think there are 37 genes here. So all of the genes that is necessary to build a functional mitochondria are not these 37 genes. So some of the genes were captured and put into the nucleus, and that made then this unable to live on its own. So, but a lot of them are in fact found here inside that structure. Not only does it contain its own DNA, but the DNA is circular. So what is the significance of that? Well, bacterial DNA is circular. In other words, it's literally built like a circle. It's like a snake biting its tail. Eukaryotic DNA is linear, and it produces these chromosomes that we talked about earlier. So when we looked at the DNA that was found inside this structure, it was DNA that was put together as a circle, which again supports the idea that at one time in our distant past this was a bacteria, because it's bacterial DNA, has the structure of bacterial DNA. And yet another piece of information is that the replication process of mitochondria, and you do and you can grow mitochondria in your body cells. So when that happens, it's fusion. It is bacterial cell division. So eukaryotic cells have a very particular type of way they reproduce themselves, and that's a double S, I believe. And bacteria have a completely different form of reproducing themselves. So it turns out that mitochondria, when you reproduce mitochondria in your body cells, they are following this form of cell division. They don't go through mitosis. They don't go through meiosis. They go through what is identified as cellular, bacterial cellular division or fusion. Actually, it's not fusion, it's fission. Thank you. Fusion is coming together. Fission is taken apart. All right. So the process is fission, and the bacteria, and that's exactly what bacteria do. So lots of evidence, and most biologists now accept the idea that this structure for sure, and many other structures probably have originally were from free-living bacteria or other organisms that were engulfed by a cell and then took up permanent residence within the cell. All right, so that's your mitochondria. Let's talk about two structures. This is cilia and flagella. Oh, before we do, I do have an image that we should look at. That's not the image I wanted to look at. This is. So I forgot to show you this image when we were talking about lysosomes. So there they are. Digestive enzymes, very simple structure, and very powerful enzymes that are capable of digesting most things that end up inside that vesicle. And then here is our typical mitochondria, and you can see that the inner membrane is dotted with enzymes because that's where cellular respiration is actually physically taking place. So that's your typical looking ribosome, and here is the image that you would get if you're using an electron microscope. Okay. All right, so we're going to talk about cilia and flagella. Not a whole lot. We do run into these. The cilia can be identified as being numerous and short, hair-like structures. On the surface of the cell, and they are composed of microtubules. We'll talk about microtubules in just a bit, okay? So these things that are on the surface of the cell are designed for the movement of substances. Movement of substances across the cell surface. So the cells are not moving, but materials that are sitting on the surface of the cells get moved, such as mucus. Mucus would be a good example in the respiratory system. In the reproductive system of a female, then the egg would be moved along by the movement of the cilia. The flagella, on the other hand, are longer and fewer in number. And there's only one cell in the entire human body that has flagella, and that is the sperm. No other cell in the human body contains this structure. Let's take a look at an image. All right, so this is going to be a cilium, a single cilia, or this singular form of cilia, I should say. And here you can see there's my microtubules. We don't have to worry about the pattern, but you can see we've got this pattern. These are microtubules, and then there's a membrane that encircles them. So the membrane of the cell extends out around them, and it creates these long whip-like structures. And as they whip back and forth, they are actually producing movement. So the power stroke is this way. The mucus that is sitting on the surface would be moved in this direction, and then it bends, whips back, and then this is how movement is provided. So they talk about movement of the cell. So that's the flagella, so we'll ignore that for the moment. So this is ciliary movement, so things like mucus and other materials that are sitting on the surface of the cell is being moved. So movement of liquid, for example. Okay. The flagella are longer. They're built in the same way, but they're longer, and there's usually a single one. Certainly in human cells, there's only one, and this would be the sperm. And this is designed to move the cell. This is designed to move things on the surface of the cell. The cilia are not designed to move the cell, but to move things that are sitting on the surface, whereas the flagella designed to move the sperm. Okay, so that's the difference between those two things. Okay. All right, we're getting close to the end here. In fact, we're almost to the end. And the next thing I want to talk about is the cytoskeleton. And I like to end the lecture on cells with the cytoskeleton, because the first thing we're going to talk about when we go into the chapter on tissues is we're going to start mentioning some of the very protein microfilaments that we are going to list right now. So the cytoskeleton of the cell is a scaffolding found within the cell Okay, and it helps the cell to organize. That is, it helps the cell to organize the interior, to hold things in place, to compartmentalize that cell. It helps the cell in the movement of organelles. Okay. It's involved in cell division. And for attachment to other cells or structures. It helps the cell to maintain shape, so it produces cell shape. So it has a number of functions. Much like your own skeletal system has a number of functions. Organizing the interior of your body, not cell division obviously, but for attachment, and also for maintaining your shape. There are 3 proteins, or categories of proteins, I should say. Now, so some of these proteins, we have different forms of them, so it's not just a 3, literally 3, but there are 3 protein types. Let's put it that way so it's easier to understand. Okay? They are in order of size, from small to large. Microfilament, and a microfilament is the smallest. And then there is the intermediate. These are proteins, by the way. I will mention that. Okay, so intermediate filaments, and obviously that's the in-between size. And the third is the microtubules. And we've already talked about microtubules, haven't we? We talked about microtubules for flagella and cilia. So that's part of the cytoskeleton of the cell. Let's go ahead and look at an image of each of those 3 different types. So 3 types. And as I said, there are variations of those types. So it's not just a single microfilament or what have you. So here, you can see microfilaments, and you can see it's producing cell shape. So you're going to be talking about microvilli, especially when you get into the digestive tract. How are these little finger-like extensions maintained? Because of the microfilaments. The cytoskeleton is allowing the cell to form that structure and maintain that structure. Okay. Intermediate filaments, we'll be talking about them when we talk about cell-to-cell attachment. So we'll look at intermediate filaments when we take a look at that. And then there's the microtubules. We've already mentioned them in terms of the cilia and flagella. In this case, they're also going to be involved in cellular division and moving chromosomes around during that process. So, ladies and gentlemen, that really finishes up our discussion of cells. That's going to be the last section, the lecture in this section. The next time you see me in this format, I will be talking about tissues. So I hope you have a good day or evening, and I will see you later.